Start recording. Hooray. Make it bigger. Yay. Oh, you can't see any? Well, that's no good. All right. Okay. DNA is stored in the nucleus in a form called chromatin. And this was page 34. Okay, so chromatin. And what is chromatin like? How can we describe it? It is long, and it is almost, we're not at noodly quite yet. It is long, and it is thin, and it is noodly. Now, if you remember, we have this DNA, and it's got this twisting ladder shape. Now, the twisting ladder actually has a scientific term for it, and that is called a double helix. Okay, a helix is a spiral. Okay, so if you hear the word helix, that means a spiral. It's a double helix because you've got one strand spiraling around the other strand. So a double helix is a twisting ladder. So we got this twisting ladder. And what it does in the nucleus is, honestly, if you just had the DNA and it was all like completely stretched out, it would be way too long and messy to deal with. So what happens is it does get packed up a little bit. Not a lot, just a little bit. So do you see how we have this little spiraling loop? And it gets wrapped around these like circular things. Now, what are these circular things? Those are some proteins. Those are called histone proteins. And why am I putting a star beside the word histone proteins? Because that's a word that you should know. So make sure that you star it. I'm sure it's on one of the vocab lists in here somewhere in this book. And I, again, have these little guys today. So when we come across a page where we need to say, oh, here's our vocab list, there's some here, there's some at the very back, there's some on that counter, and there's some on that counter so that you can come and get them when you need one, okay? However, it still coils up even a little bit more, and once it is coiled around, hello? Yeah. Yeah, sure. Once it's coiled up even more, then it is called chromatin. Okay, so you got the DNA, it coils up a little bit, it wraps around those histone proteins, and then it is called chromatin. It's still really long. It's still really, really thin. It's just not quite as long and quite as thin as it would be otherwise. Okay? Now, how many of these pieces of chromatin do you have in one cell? Does anybody know? You have 46 of these pieces of chromatin, 46 strands of DNA. Okay? In every cell. Oh, why am I writing this down? It's right over here. If you would like a highlighter and you don't have a highlighter, remember there's a box of them at the back of the class. Okay, and you can feel free to help yourself to one. So there are 46 strands of DNA. In other words, there are 46 strands of chromatin. And do you know where they came from? 23 of them came from your mom, and 23 of them came from your dad. Okay? So there's 46 strands of DNA, 23 came from mom, 23 came from dad. Have you guys heard of the company called 23andMe? Okay, it's like a DNA, it like analyzes your DNA and it's supposed to tell you where you're from, right? I don't know how good of a job it really does at doing that, but that's where the 23 comes from, is that 23 of your chromosomes, you have 23 pairs of DNA molecules, one from mom and a matching one from dad, okay? Now, here is a picture of the nucleus. So you can imagine that in the nucleus, there's our chromatin. It looks kind of long and thin and noodly, maybe like a piece of yarn or something that, like if you took a thing of yarn and you know, like a ball of yarn and you just, you know, undid the ball of yarn and you threw it on the ground, that's what the chromatin would look like, okay? What do we call that thing in the middle of the nucleus that's kind of dark? That's called the nucleolus. Nucleolus. Does anybody remember what the nucleolus does? It makes something.
It makes, what are they? Ribosomes.
It makes ribosomes. I mean, this whole thing is like a star, right? Now, in the drawing, you can see the chromatin. But if you are actually looking at this, even under an electron microscope, okay, and do you guys know about electron microscopes? Electron microscopes are special microscopes, and they can see, you guys remember using the microscopes last year? And you could see things like 100 times bigger than they were. Electron microscope can go to like more than 1,000 times bigger, okay? So they can see things that are very, very small. What does the chromatin look like in reality? It looks like absolutely nothing, okay? In reality, chromatin is so thin you cannot even see it, all right? Even under a microscope, you can't even see it. But what you can see is you can see the nucleolus, yes? So that's a nucleus with the nucleolus in the middle there. All right, now, in order to get bigger, right, if you are still growing, some of you are probably still growing, some of you may have stopped growing. I stopped growing when I was in grade 8. I think I was 13, and I got to 5'2", and I never made it any taller. I was like, oh, shucks. But what can you do? Here I am, little short me, okay? But in order to get bigger, did I just have one cell that got bigger and bigger and bigger and bigger and bigger and bigger and bigger? So I'm just one big blobby cell? Or what else could happen to make me bigger? I could take one cell and I could make a copy. So I have now 2 cells. So when an egg and a sperm get together, it makes 1 cell. Yes? I know. It's so funny because we're 14. Okay, egg, sperm, 1 cell. How long should I give you guys to be amused by this? A little while, we're good? We're good? Okay. Okay, and this 1 cell, we're not going to actually talk about this year, but this is, you'll talk about it next year. This is called a zygote. Zygote. What a great name is that, hey? So when the egg and the sperm fuse together, you get a zygote, and it is a single cell. Now, if you were to say a single cell, you would just be a big blobby cell with like a nucleus and a nucleus. That would be ridiculous, okay? So that cell then makes a copy of itself and it divides into 2 cells. And those 2 cells divide into 4. And then those 4 cells divide into 8. And then they keep dividing and keep dividing and keep dividing until you are a baby human, okay? And then anytime that you need to grow, your body will divide some of its cells to make new cells. And then those cells will get a little bit bigger and then they will divide again. And then they'll get a little bit bigger and then they'll divide again, okay? Just before a cell divides, the chromatin actually coils up even further. Do you remember how the chromatin was like that ball of yarn that I'd unraveled and thrown on the ground? Well, before the cell divides, we're actually going to take all of that messy yarn and we're going to ravel it up into a nice ball of yarn. Yes? And why is that? Because we need it to be easy to organize. Because if we're dividing the cell, can you imagine if you had 46 noodles and then you made a copy of all those 46 noodles? Now, how many noodles do we have? 46 and 46, that would be what, 92 noodles? Okay, so imagine that we have 92 noodles all mixed together, and we need to get half of them over to one side and half of them over to the other side so that each new cell can have 46 noodles. Can you imagine how difficult that would be? Wouldn't it be nice if instead of being all long and thin and noodly, they were all packaged up nice and tidy? So that we could be like, oh, you go over here, and you go over there, and you go over here, and you go over there. So you just have balls of yarn that you're sorting instead, okay? And what that is before a cell divides, we say that the chromatin condenses so the chromatin coils up into a chromosome shape. So the chromosome is like the ball of yarn. We say it is condensed. If something is condensed, you pack it up tight. Does that make sense? Okay. Now, if it's unraveled, the chromatin fiber, this would be like the unraveled ball of yarn, messy. In other words, it's long and thin and noodly. So the chromosome is the packed up tight version. The chromatin is the long and messy version. So basically what happens is we start with our DNA. What do we call the shape of that DNA? That shape is called a double helix. Okay, so we start at the most simple level as a double helix. And then the DNA wraps around, what did we call these things here? Those are called histone proteins. And I'm going to put this as an enriched thing. If you want to know the term beads on a string, that's That's what it looks like. Does that look like beads on a string? Like you've got a string? And these would be our beads. Does that make sense? You're like, I don't know. Does it make sense? I'm falling asleep. I don't know. All right. So we've got our string. We've got our beads on a string. It coils up even further. And we get the chromatin fiber, which is still long and thin and messy. But then it coils up. This is the most coiled up. And the thing is, by the time it gets to that point, it actually literally does look like an X. And if you look under a microscope, you can actually see an X. Now, sometimes the X's are kind of smushed a little bit. So they'll be like, right? So they don't look perfect. They won't look perfect like this one here. But you can actually see them because it's not so thin anymore. Yeah? Yes?
How exactly does it form?
How does it get coiled up like that? That is a very good question. I think and I haven't studied this in like forever, like 35 years probably. I think it has to do with, there's these proteins in the cell called microtubules, and they are like, their job is to attach to things and move them around. So I believe that's how it happens. And then there's going to be intermolecular forces in between them. And there will be like hormones that signal, oh, this process needs to happen, and it will turn on certain genes and it'll make proteins that cause the process to happen. That is the generic overview. It's very vague, and I don't know if I'm correct, but that's my best guess. Okay? So this is going to allow for easy organization and movement of DNA molecules during cell division. Okay? It also makes the DNA very stable and very compact and less susceptible to damage, right? Because not as much of it is exposed. So we have chromatin, and then we have chromosome. Chromatin, chromosome, chromatin, chromosome. You guys got to do this with me now. Chromatin, chromatin, chromosome, chromatin, chromosome. And you know what? The people who have done this with me are actually going to remember it. Yes? And even though you get a cheat sheet this year, if you remember it this year and you're like in grade 11 and you're like chromatin, chromosome, and you're like, I didn't need to study this because I already know it, because I've done an interpretive dance. Yes? And just in case you didn't believe me that you can actually see these, this is under an electron microscope. You can actually see the three-dimensional chromosomes. Okay? Now, on this page, there's going to be a little bit of enriched stuff. Most of this is fair game for everyone, but there's a little bit of enriched stuff as well. Step number 1. This is a normal cell doing its cell business. This is a normal cell doing its normal cell thing. If it's a heart cell, it's helping to pump the blood. Yeah, if it's an eye cell, it's helping you see. If it's a skin cell, it's helping protect your body. If it's a nerve cell, it might be helping you feel something or transporting signals or whatever. So that's just a normal cell doing its normal cell thing. And during that time, the chromosomes are not chromosomes, they are chromatin. Okay? And this is the enriched part. This phase of a cell's life is called interphase. Interphase. And that's when we have the normal cell just doing its normal job. Okay? And what do you guys remember what inter means? Inter means between. And in this case, phase just is what we use at the end. It's actually in between cell divisions. And can you imagine trying to organize this mess? No. No. So that's number one. So where does a cell spend most of its life? It spends most of its life in interphase, doing its normal cell job, doing its normal cell thing. However, sometimes it's going to be like, oh, I'm an old cell. Maybe I need to divide. Maybe you've got a cut on your finger or something like that, and it needs to heal. So you need to make new cells to heal over that little cut on your finger or whatever. And so we need to start organizing those chromosomes. So the chromosomes start to condense. What does it mean to condense? To condense means to coil up. Okay, so you need to know the word condense. You need to know the word chromosomes. Okay. This is actually under just a regular microscope, like the kind that you would use in class. Okay. And you can see that we have what can start to be looking like little thread-like things. Yes? But they're actually a lot thicker. So these are condensing into chromosomes. This phase here, this is called prophase. Pro means at the beginning. I spelled beginning wrong. That's okay. So cell division starts here. Okay, so we get prophase at the beginning of cell division. And now we end up with the chromosomes being fully condensed into their X shapes, but you can see they're still sort of all mushed on top of each other and whatever. Yes, it's like the yarn is in a pile in the middle of the store. Yes, and you need to sort it out. Yeah? And this is still prophase. So that's number one. This is number two. This is number three. I want to make the numbers bigger so you can see them. Okay, so this was one. This was two. This was three. And then number four, the chromosomes line up along. Yeah. Number four, the chromosomes line up along the middle of the cell. Now, this is an actual cell. And this is a diagram, right? Because does the actual cell look beautiful and easy to understand? No. So that's why we have a diagram, to make it a little more beautiful and easy to understand. And you'll notice that we've got half of the X on one side of the cell and half of the X on the other side of the cell. I want you guys to hold up your arms like this, half and half, Because what's going to happen is that when the cell begins to divide, these two halves are going to separate. Okay, they're going to separate. Those two halves are actually identical. Okay, so this right hand is, my right hand is a copy of my left hand. They are identical, so you're getting a full copy going to each new cell. Okay?
Wait, no, there's like a mutation from like one cell to another.
Mutations usually will happen, it happens in one of the cells. When the DNA makes a copy of itself to get ready to divide, it will actually make a copy of the mutation, if it's a mutation that has stuck around. Okay? Now, to be honest with you, we are all mutants. All of us have mutations. Otherwise, if we didn't have mutations, we would all look and be identical to each other. Every single thing, from your hair color to your eye color to your height to whether your finger bends backwards or not. Some people's thumbs do not bend. Yours bends way backwards. Okay, mine bends backwards, but not quite that much. Some people, try it out. See if your thumb will actually bend backwards. This is called hitchhiker's thumb. Okay, that's just the name they give for that. It's like saying you have brown eyes. Okay, you have hitchhiker's thumb. Okay, this is non-hitchhiker's thumb. So if you cannot make it go backwards, that's non-hitchhiker's thumb. There is one gene. There is one gene in your body on one chromosome that says whether you're going to be able to do this or be able to do that. And that would be the result of way back sometime, somebody had a mutation. Probably it started out like this, and then they got a mutation that allowed it to do that. Right? All right. This phase here, where the chromosomes line up in the middle, that is called metaphase. And then if you remember, the two halves of the chromosome get pulled apart. Okay? So these guys get pulled all the way. And again, there's some little microtubules and proteins that are pulling them towards the ends of the cell. And this is called anaphase. Pardon me?
Is this a cell?
Is this a cell or a...
Or is this a cell or inside of a nucleus?
Oh, this is the entire cell. This is an entire cell. Because actually what happens is that in order for the cell to divide, you'll notice that here we have a nucleus, yes? Here we still have a nucleus, but there's no membrane anymore. Okay, and then by the time we get here, where's the nucleus? The nuclear membrane breaks down to allow the separation of those two chromosomes, okay? So that's anaphase. And then finally, all the chromosomes end up at either one end or the other end of the cell, okay? And that's called, sorry, that shouldn't be an I. I was trying to underline a T and it ended up looking like an I. This is called telophase. So we end up with once the two chromosomes are pulled apart, and now we have 46 chromosomes over here, and we have 46 chromosomes over here. That's when, yeah, sorry.
Wait, what if you have not enough or too many chromosomes?
What if you have not enough or too many?
Yeah.
Is that what you're asking? Yeah. That will result in genetic disorders. Okay, so like if you have an extra, is it chromosome 21? That would cause Down syndrome. Yeah? So for example, you'll end up with Down syndrome, often depending on the chromosome. They call it trisomy because you have 3 copies of chromosome 21. Anytime you have 3 copies of a chromosome or you have only 1 copy of a chromosome, either you're going to end up with some sort of genetic disorder, or it's going to make it so that the cell actually can't divide properly, right? So you have that, you remember the zygote, right? The egg, sperm, they come together, you get a zygote, okay? And what'll happen is that the code for how the DNA code for how the cells divide doesn't work properly. And so that will actually end up in a miscarriage because it won't be able to develop past a certain point because it's not dividing properly, okay? So miscarriages, as tragic as they can be, are not always, and that's not the only cause of a miscarriage. You can just have miscarriages too because there's not proper adhesion of the fetus to the placenta, right? There's other causes, but definitely if you have genetic abnormalities, it will eventually undergo a miscarriage. And many women actually have pregnancies that they don't even know that they were pregnant because it happens so early on, right? And they just think, oh, my period was a little bit late and now it's heavy, right? So that can definitely happen. Yes?
I can't remember exactly which chromosome it was.
Yeah.
But there was a certain one where it like
Yes.
and it could cause like if you lose like two sides, you get ring chromosomes, like that kind of thing.
Yeah. So you can also lose, in addition to having like only one of a particular chromosome, some chromosomes can actually have little bits come off of them. And again, those cause certain disorders, okay? However, some of the disorders are, they don't actually cause huge problems, right? And some of them do, right? So it just depends on which one and how much is missing. And sometimes you can get like a transposition where a little bit of this chromosome ends up sticking onto that one, right? So there's all kinds of things that can happen. In addition to just single changes in single letters of the DNA code, usually those single letter changes are the ones that don't actually cause huge problems. And those are the ones that'll do things like change an eye color, right? Or make the hitchhiker's thumb, or decide whether your first knuckle has hair on it or not. That's another one. You can look on your knuckle and say, is there hair on there? If so, then you have the hairy knuckle gene on the first knuckle. If you don't have hair on the first knuckle gene, then you have non-hairy knuckles. I think everybody has it on this first one, but not everybody has it on the one that's closest to your fingertips. This is all, next year you guys get to do a deep dive on all of the genetic stuff. That's where this is going for grade 10. So hopefully, if everything goes well, you end up with 46 chromosomes on one end of the cell, 46 chromosomes on the other end of the cell, and then the cell membrane starts to do what? How would you describe this? It pinches together in the middle, and then it separates into 2 new cells, each with 46 chromosomes. Yes? What causes it to pinch?
Like obviously it's not like that.
Yeah, no, it's actually called a contractile ring of proteins that go around the cell, and they kind of like just tighten and tighten and tighten and tighten. So they would like go like this. They slide across each other, and so the whole thing becomes smaller and smaller and smaller around the middle until it pinches off. Okay, so they pinch apart into two new cells, and this is called cytokinesis. We are not really going to talk a lot about mitosis this year, but mitosis is a grade 9 topic, okay? And all mitosis is it is the cell division part of the cell life, the cell cycle, we call it, okay? So the cell cycle it goes interphase, mitosis, cytokinesis. Interphase, mitosis, cytokinesis. And then, once it's undergone cytokinesis, that cell can then grow, and it can undergo mitosis and cytokinesis. And then that one can grow and do its cell job and do all that stuff, and then it can undergo mitosis and cytokinesis. And on and on and on. So what is this mitosis thing? Mitosis is made of prophase, metaphase, anaphase, and telophase. And do you know what that spells? It spells PMAT. PMAT. And you know what happens before mitosis? You do. It's right here. What happens before mitosis? Interphase. So the way I remember it is kind of embarrassing, but I pee on the mat. So you have interphase, and then you have prophase. So I pee on the mat. So if you're into this kind of thing, if you're thinking biology is my jam, and I love this, I would recommend like kind of just being familiar with this. Okay, it is enriched. I'm not going to test you on it or anything like that, but it is interesting. Okay, and if you're into biology, and if you really want, do you guys want to learn a song? Okay. Okay, so interphase is actually made of 3 phases. They're called G1. Interphase, there's G1, where the cell grows. There's a phase called S phase, where the DNA makes a copy of itself. Yeah, because how are you going to end up with 2 copies to move to either side of the cell if you don't make a copy first? Does that make sense? Okay. So G stands for growth, sort of. Okay, so basically the cell is growing and doing its cell job. S stands for synthesis. Synthesis means making. Ooh, okay. And then there's G2, which is more growth. Okay, So the cell gets bigger, the cell does its job, the cell gets bigger, the cell does its job. It makes more organelles because as it gets bigger, it needs more organelles, it needs more ribosomes, it needs more endoplasmic reticulum, it needs more stuff. So that's what's happening during the G. But during that S phase, it's making a copy of the DNA. So the DNA is like, I got one strand, right? I got 46 of them, and they all replicate. So I have my unreplicated DNA, and then I have my replicated DNA. They all make a identical copy so that when they separate, you have a copy going to cell A and you have a copy going to cell B. Yes? And our little song actually sings us through the entire cell cycle in order from G1 to S to G2 to prophase, metaphase, anaphase, telophase, and then cytokinesis. All right, so here's how the song goes. And okay, this is a cycle, yes? It goes around. This song is a round. Oh my God, it's a round, and it's a beautiful round. It's actually a Christmas song. I can't remember exactly how the Christmas song goes, but I do know this version of it. So it goes G1, SG2. Okay, so you guys are going to sing with me. G1, SG2. All right, we'll try that again. G1, SG2. And then it goes prophase, metaphase, anaphase. Prophase, metaphase, anaphase.
Oh, is that Rose Rose Red?
Could be.
I know that song. It's
Okay.
around me doing choir.
Ah, there you go. Except for now you've got like biology lyrics to it, okay? And I've started too high because I can't go that high. Okay, so I'm going to start a little lower. G1, SG2, prophase, metaphase, anaphase. My voice is going weird today too. Telophase and cytokinesis. G1, SG2, prophase, metaphase, anaphase. So let's just do that part. G1, SG2, prophase, metaphase, anaphase. Let's try that again. G1, SG2, prophase, metaphase, anaphase. And then we go up high, telophase. Okay, so telophase. From the beginning, G1, SG2, prophase, metaphase, anaphase, telophase. And then we go, and cytokinesis. So let's just try that part. Telophase and cytokinesis. Telophase and cytokinesis. G1, SG2, prophase, metaphase, anaphase, telophase and cytokinesis. G1, SG2, prophase, metaphase, anaphase, telophase and cytokinesis. G. Now, you've done it as a round. Do you want to come in?
Wait, what?
Do you want to come in on the second part? Okay, so I'm just going to let you do your thing, and if you end up singing with the Rose Red lyrics, that's okay. Okay? Anyone? And do you want her to join you?
Join me in the end.
Okay.
Yeah.
So I'm gonna go. All right, and then these guys are gonna come in with the round. So if you guys can sing with me, that would be very helpful. Okay?
We could.
G1, SG2, prophase, metaphase, anaphase, telophase, cytokinesis, G1, SG2, prophase, metaphase, anaphase, telophase, and cytokinesis, G1. Okay, and you can actually have three people. You can have people come in after that as well.
We did it in like a
Yeah, so it's kind of crazy. Anyway, thank you for humoring me. I haven't done that in years, so it made me kind of happy to be able to do that. Not every class, I cannot get buy-in from even 4 people to sing along with me some years. So that made me very happy. I appreciate it, no end. And could you please take that out? Thank you. And put back whatever it is that you took. Thank you. All right. So every organism has a certain number of chromosomes. Chromosomes are always found in pairs. Okay, so it doesn't matter whether you're a human or a baboon or an apple tree or a fungus or a clownfish. Doesn't matter. Your chromosomes always come in pairs. One member of the pair came from mom. One member of the pair came from dad. Most human cells have 46 chromosomes arranged in 23 pairs, including one pair of chromosomes that helps determine the sex. Now, it's way more complicated than this, but we're just going to go through the most simple of sex chromosome pairings. Either you have an X chromosome and a Y chromosome. And if you have an X chromosome and a Y chromosome, that means you are at least genetically male. And if you have an X chromosome and an X chromosome, at least genetically, you are female. But as we all know, it is way more complicated than that, but that is at its most simple state for humans, okay? Fruit flies only have 4 pairs of chromosomes because fruit flies are a lot simpler than we are. Yes? So here is a picture of all the chromosomes of a human. Here's a picture of all the chromosomes of a fruit fly. Is this human genetically a male or a female? Genetically, this is male. How about this fruit fly? Genetically, female. Now, we're not going to be spending a whole lot of time on this, but you should be aware that grade 9 presents only the most basic view of chromosomes and sex and does not deal with specialized situations, for example, like being intersex. We're not even talking about gender identity here. We're talking about literal differences in how the body, the chromosomes in the body, and how those chromosomes are activated or not activated. There are many, many people in the world who are actually intersex. And what that means is that they either have genitals, sorry, that was supposed to be a highlight, not a cross out. They either have genitals or chromosomes or reproductive organs that don't fit the male-female binary, okay? So for example, their external genitals may not match their reproductive organs. There could be one thing on the outside and another thing on the inside. And they wouldn't even know necessarily unless they have like some surgery done or something like that. Do you know what I mean? You're like, I see what's going on down there. I'm pretty sure that's what it is. And then all of a sudden, you know, you get a surgery and you're like, oh my gosh, there's a uterus in there? Good Lord. Right? And how would you know? Right? Pardon me? It depends on what version of intersex there is. Okay, so there's many, many different versions. They may have traits, they may have traits of both. Being intersex is not a disorder, it's not a disease, it's not a condition, it is just one of many different genetic ways of being a human being. Yes? And we have here a little video about what it is like to be intersex, and we're going to watch that for just a moment. I have to go and grab the actual link because I forgot to link it this morning. Okay.
Raise your hands if you have testes. I'm Pigeon.
I'm Alice.
I'm Emily.
I'm Cypher.
And we are Intersexy.
Okay, we're going to just pause for a second because I cannot make it full screen apparently. Why is it not going full screen?
Whoa.
How do I, where, why? Ah, there we go. Okay. There we go. Okay, we're going to put on subtitles.
Raise your hands if you have testes. I'm Pigeon.
I'm Alice.
I'm Emily.
I'm Cypher.
And we are Intersexy.
Intersex describes a person who doesn't fit the typical definition of male or female.
I have XY chromosomes, but typical female genitalia.
I'm a girl who has testes and XY chromosomes.
I identify as a queer, gender non-conforming intersex person.
I identify as a Black intersex man.
Intersex is not new. It's been around since the beginning of human existence. I mean, there's probably even intersex dinosaurs, if you think about it.
Transgender has to deal with your gender identity, whereas intersex has to deal with your biological characteristics.
Often, intersex people get surgeries that they don't want, and transgender people have to fight for surgeries that they do want.
So what'll happen is you might have someone who is born intersex who ends up getting a surgery when they're very, very young, because especially this used to happen a lot in the past. People would be like, oh, you know, this is a girl and she's got like a testes, and so let's get rid of those. And they'll do it before like the kid is even like old enough to have a say in it, right? Because they're like, we must make people fit whatever the normal mode is, right? So hopefully that's not happening as much anymore, but it still does happen, I'm sure. They
gave my mom the excuse that the internal testes were cancerous, that I would develop cancer.
They didn't even
They
come up
weren't.
with an excuse, basically, in terms of a health-related reason. They instead just said it was about the appearance.
A lot of doctors are very uncomfortable with the idea that I have testes, and they're still trying to get them removed, but I'm perfectly healthy and there's nothing wrong with them.
They did a surgery to remove my testes and told my parents to take me home and just raise me as a girl, and I didn't find out about it myself until I was 12.
There aren't a lot of options, or medical providers don't explore other options. My mom would put me in dresses and she would be like, oh, aren't you so cute and you're so pretty? And I would be like, no, this is horrible. I was put on hormonal treatment, which consisted of estrogen and progesterone. I just wanted to belong. I wanted to fit in. I didn't want to be different. So even though I knew something felt amiss, I conformed. He was very condescending. He was like, you intersex activists don't know what you're talking about.
It's difficult for intersex people to find each other because from an early age we're told not to talk about our bodies.
I did feel like I was the only one.
My doctors always told me there was nobody else like me, and so it just perpetuates a vicious cycle of shame and stigma that we can't break out of. I would tell another intersex person that you are worthy, you are lovable.
Your body is beautiful, you're beautiful.
Intersex people don't need to be fixed. There's nothing wrong with them.
I know you feel like you might not be able to get through this. I know you might have really dark thoughts, but I want you to know that meeting other intersex people and finding a community or a support group can be one of the most important aspects in your healing process. And we're out there, we're out here, we're here. And I just hope you can find us.
Okay, so just to be clear, this is different from gender identity because we actually have biological differences, okay? And we're not going to take all the time to read through the entire article here, but I'm just going to go and take a look. If you're interested in reading the entire article, the link is in your book along with a QR code. And it talks about being intersex. It can affect the genitals, it can affect the chromosomes, it can affect hormones, the reproductive system, and it can affect the gonads, which is the generic term for the ovaries and the testicles, okay? So just like genitalia is the overall term for the, external sex bits, the gonads is the overall term for ovaries and testes. So what causes it? There can be a change to an androgen receptor gene. There can be natural or synthetic hormone exposure during embryo development. There can be missing or out of place sex determining region on the Y gene. So that would be one of those things where something maybe is missing in a chromosome or is out of place. And there can be other genetic conditions that cause hormonal abnormalities that just make people develop differently. You can have different combinations of chromosomes. You can have instead of being XY or XX, some people have XXY. Some people have some cells in their body that are XY and other cells in their body that are XX, right? You might just have one X chromosome and no Y chromosome. Unfortunately, you can't just have one Y chromosome without an X chromosome because the Y chromosome is actually a little smaller than the X chromosome, so it's missing some key bits that you need an X chromosome for the cell to work properly. If you have two Ys, again, you're going to be missing stuff. You can have an XYY, but I don't think you can just have YY, and at least that's my recollection. It's been a while since I've studied this in depth, so I could be wrong. Okay. Is a person who is intersex a hermaphrodite? No, hermaphrodites don't exist. This is an outdated term implying that a person is both fully male and fully female, which isn't biologically possible. In fact, many people who are intersex consider the term derogatory and stigmatizing, so just make sure that you don't use that term out in the big wide world. Do they need surgery or treatment? You typically do not require any surgery. Some people will choose to have surgeries, but that has to be with their own informed consent. There are risks of intersex surgery: decreased sexual function, infertility, scarring, urinary incontinence. Do you guys know what that is? It means where you don't have control over your bladder, so you'll just pee yourself. Yeah? Okay. So, and urinary tract infections. Are there health conditions associated? Some people do end up with osteoporosis. And that's just because some of the genes are linked with other issues as well. Let's see, hypospadias. The urethral open where urine leaves the body is on the wrong side of the penis, or there is no urethral opening. You can have Klinefelter syndrome where there's an extra X chromosome. You can have Meyer-Rotten-Kansky-Kuster-Hauser syndrome. Wow, that's a lot of names. That's where the uterus and vagina don't form as expected. You can have Swyer syndrome where you have undeveloped sex glands. How do you know? You might not get puberty. You might have changes in puberty that don't align with whatever your sex was recorded as, like males growing breasts or something like that. You might have fertility problems. Does intersex being intersex affect fertility? It is possible that it might affect fertility. And so many people can have assisted reproductive technology like IVF, donated eggs, donated sperms, things like that. And anyway, if you want to read more about that, there's a lot online. I find it very fascinating, actually. It's a really interesting topic. But at any rate, today, what you are going to do is you are going to do pages. What happened here? Oh, that's right. That's because this is this one. I want my workbook. There we go. Today, for homework, you are going to do pages 38 up to 40. I would also like you to start on your cheat sheet, if you have not already. And if you would like, actually, there is a vocab list here on page 38. So if you want a little sticky note to mark page 38 for yourself, please help yourself to one of our little stickies that's on page 38. Okay, there's stickies here, there's stickies on the side counter, stickies on the back counter, stickies on the other side counter. That's page 38. So page 38 and page 38 and page 8. A good place to start on your cheat sheet would be the vocab on page 38 and page 8. But you can also be drawing diagrams that we've seen and things like that. Like, it's up to you what you want to put on there. But Like I said, the vocab on pages 38 and 8 is a good place to start. Okay? Am I having fun?